Okay, in this video I will define phylogeography, and then we'll also talk a little bit about what markers are used in today's studies and how that compares to what they used to use in the past, hence evolution of phylogeography datasets. So first, before I give you a definition, I would like you to think about it based on if you've already done the reading, or if you're just looking at the name, phylogeography, or you can look at this picture here, which is a graphical representation of what is commonly done in phylogeography studies. So I'll give you a little bit of time. You can think or jot down some notes. What do you think phylogeography is? Okay. So really, the goal of phylogeography is to understand how historical events have shaped today's distributions of genes, populations, and species. So in the previous species module, we talked about molecular ways to identify different species. Here with phylogeography, we're moving on to, you can think of the next step in that we're now comparing differences among species. And if we look at the name here, if we break down the word phylogeography, the philo part here means groups having common ancestry. And then geography here is different locations. So what phylogeography is interested in is how the ancestry of different species or different populations is related across their current different geographic locations. But the unique thing about phylogeography is that we're also interested in this historical aspect. And what do I mean by historical events? Often things that people doing phylogeography studies are interested in are things like ice ages or drifting of continents, just like what's shown here in this movie poster. So when we're thinking about phylogeography, again, we're trying to figure out how historical processes may have shaped the current distributions we see of species across different geographic locations. So really, phylogeography embraces these aspects of both time, which is time in the sense of evolutionary relationships, and space. Space being geographic distributions across different geographic sites. And so really, by comparing evolutionary relationships of genetic lineages to their geographic locations, what we can do is understand which factors influenced their distributions of genetic variation. So that's really what we're interested in. And if we go back to this same diagram, this is a tree. We'll talk about that later this module, but this is a tree that I'm showing. So if we think about how this tree is demonstrating these different aspects of phylogeography, we have this time aspect here, which is these evolutionary relationships. So those evolutionary relationships are demonstrated by the branches on the tree. So we start here. This is considered a common ancestor. Each one of these tips is, say, a different individual. And so we can see that these individuals that were collected from the Red Sea all are relatively similar to each other. They're all clustered together. They're close together on the tree. The longer the branch length is, that's indicating this evolutionary time. So these two individuals here are more closely related. They have fewer evolutionary distance between them compared to, say, this individual and this individual. We'll talk more about trees, like I said, in an upcoming video, but overall, these distances on the tree represent these time aspect of evolutionary relationships, and then the space aspect comes in where you're then mapping these different individuals to where they came from geographically. So that's phylogeography. And to build on some of our previous knowledge, previously we've talked about different types of molecular markers, so I want to give you a little bit of time to refresh your memory here and think about what type of molecular markers might be best for phylogeography. And first, I just want you to think about what are the options? What different types of molecular markers are there? So I'll give you a little bit of time to jot that down or just think about it to yourself before I continue. All right. So until recently, the most common phylogeography markers were mitochondrial and chloroplast genomes. And if we think about this, there are pros and cons to using these types of markers. So if we start with the pros, so these are good things, there are universal primers available, which we've talked about before. So if there are universal primers available, that means it's relatively easy to actually conduct these type of studies because there are primers available that will work for a broad range of taxonomic species. So again, an example of universal primer is there's primers available to amplify all fish species, for instance. So that's a positive to using these types of mitochondrial and chloroplast genomes as phylogeography markers. But if we think about cons, reasons why it's not so good to use these types of markers, what are the inheritance patterns of mitochondrial and chloroplast genomes? Are they uniparental or biparental? Right, they're uniparental. And so what that means is if we're only using these markers for our phylogeography studies, it could lead to an inaccurate, incomplete picture of what's actually going on. So remember, mitochondria, for instance, are only maternally inherited. If we're looking at species such as sperm whales, where they know that males and females have different dispersal patterns, if we're only using a mitochondrial marker for our phylogeography study, we're not capturing the entire picture of what's happening because we're not using a marker that captures both what's happening for the males and the females. So because of these cons here, now it's actually more common to use multiple loci across genomes for phylogeography markers in phylogeography studies. And some of the two most common ones that are used now are microsatellites and SNPs. So this is a refresher showing these two types of markers side by side. Remember, microsatellites are repeated bases, so we have these two bases AC repeated a different number of times, and a SNP, there should be an E here, single nucleotide polymorphism, is a single base pair change at a specific point. And this is just one really zoomed in portion of the genome, but remember, if we extend our genome out here, there are also microsatellites and SNPs at many different points throughout that genome. So you're able to get multiple loci at many different places throughout the genome if you're using SNPs or microsatellites. And if you're wondering, okay, you can use SNPs or microsatellites, but which one's better? They've actually done several studies to directly compare the different types of markers. So this is a study that was done here in California in the San Joaquin Desert. This little guy is a blunt-nosed leopard lizard. And what they were doing in this particular paper, which is referenced here in their phylogeography study, they were looking at range-wide population structure. We'll talk more about that later this semester, but that's basically trying to see if different populations of this lizard at different specific locations within the desert have different genetic differences. And what they were looking at specifically in this paper was seeing if changes in land use throughout history affected how these different lizard populations were connected or not connected. And so what they did is they used several different types of markers to ask these same kinds of questions to compare and see which one might be best. So they used 3 different types of markers in this study. They used SNPs that were identified from RAD-seq, which remember is a type of high throughput sequencing. And they also used microsatellites and mitochondrial DNA. And overall, they found that SNPs were more informative than microsatellites or mitochondrial DNA. So this is telling us in general, if you have an option, so if you have the funding, usually that's a limiting factor. if you have the funding available to do a type of RAD-seq study, SNPs are going to give you better information than microsats or mitochondrial DNA. There are also several other examples directly comparing different types of markers in your textbook if you're interested in more examples. So overall, SNPs seem to be better, but there is also some evidence that indicates if we're interested specifically in recent patterns of dispersal, microsatellites might be better. So remember, microsatellites we've talked about previously are highly polymorphic, which means they have a high mutation rate. And so because of that, we've talked about previously how microsatellites are really good for differentiating individuals of the same species. And so what they did in this study that's referenced here is look at seagrass populations in the Indo-Australian archipelago. I wish I could have done this study so I could have gone to those places. But what they were looking at, again, is this genetic structure, so whether different populations of the seagrass have genetic differences, and they were looking at whether those patterns matched either contemporary ocean currents, so what we know ocean currents are as they are occurring today. Ocean currents are often one of the main ways that things like seagrass are transported between locations, or are these genetic patterns more matching to things that happen historically, like Pleistocene sea level fluctuations. And so they also in this study used different types of markers. They used SNPs, they used microsatellites, and what they found was that when they used microsatellites, those data seemed to reflect these recent patterns of dispersal that would be happening from the ocean currents. The current ocean currents. That's confusing. Contemporary ocean currents. So again, overall, SNPs seem to be best, but if you are specifically interested in looking at individuals that may have dispersed very recently, microsatellites might be better for that type of info.